Perfect. Okay, so now we'll continue with the purine and pyrimidine synthesis. Okay. Um, to maximize the use of number of ATPs in your brain, you need to have one pen and paper and keep writing whatever you see on the whiteboard. Okay, that's going to maximize. Otherwise, we may, even I was zoned out after reading one hour of pyrimidine and purine synthesis. So we'll try to begin from the basics again. Okay, so we are going to do purines and pyrimidine synthesis. Now, quickly remind me, purines, how many rings?
Two.
Very good. Purine has two rings and pyrimidine has one ring. And can you name some purines for me?
Uh, A and G means adenine and guanine.
Perfect. Pure as gold, adenine, guanine, and pyrimidine. Name me.
Cytosine, uracil, and thymine.
Wonderful. So CUT, cut the pie. That's pyrimidine. Okay, now we are going to talk about purine and pyrimidine synthesis. Now, if I say adenine, guanine, cytosine, uracil, thymine are purines and pyrimidines, what are they? Are they nitrogenous base, sugar, or phosphate? What are these? Nitrogenous base, right? They are nitrogenous base. Nitrogenous base. So, adenine, guanine, cytosine, uracil, thymine are nitrogenous base. That means it has nitrogen in it. Okay. You remember two ring structure and the one ring structure. So they are nitrogenous base. Now, I just want one sugar. Can you name me one sugar which I can attach with the nitrogenous base? What kind of sugar should I attach? Name me just one sugar which I can attach to the nitrogen.
Deoxyribose
Deoxyribose
uh,
or
or ribose.
just ribose, right? Um,
Okay.
organic chemically speaking, it's ribose 5 phosphate. Okay, and that's our sugar. Nitrogenous base, ribose 5 phosphate is our sugar. And quickly tell me, how can we form ribose 5 phosphate? $10 question.
Add phosphate.
Uh, how, what cycle can form ribose 5 phosphate? Starting from
Uh,
P,
purine ring.
No, pentose phosphate pathway.
Okay.
Is also
Okay.
called as, which is also called as
Efficient.
HMP shunt. Wonderful. So now you know how can we form our sugar from pentose phosphate pathway or HMP shunt. Keep writing this, okay? Remember, you need pen and paper to engorge all these things in your mind, okay? So we have nitrogenous base, we have one sugar. If I combine ribose 5 phosphate and nitrogenous base, what do you form? What will this form? Sugar and nitrogenous base. I'm going to combine them. What is it going to form?
Nucleoside.
Very good. Nucleoside. Perfect. Now the name will say, okay, what's the nucleoside of adenine? Quickly. Brr. Adeno, scene, guanine, it's guanosine. Similarly for cytosine, what's the nucleoside?
Cytosine.
No, cytosine itself is the name. Uh,
Cytodine.
cytidine.
Cytodine. Okay.
Cytidine. What's this? Uridine. And finally, we have
Thymine.
thymidine. Perfect. So now you made nucleosides. I just want to add one phosphate and you can make what? So nitrogenous base plus sugar, that's nucleoside. And I just want to add phosphate and then entirely you can make what?
Nucleotide.
Nucleotide. Perfect. Nucleotide. And this can be depending on the number of phosphates. If it's just one phosphate everywhere, adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, UMP, TMP. Okay, is it clear to everyone what's nucleoside, nucleotide? Now what am I going to do here is we are going to do purine and pyrimidine synthesis. Now these are purines and these are pyrimidines. We will make them. Okay, we are going to make AMP, GMP, CMP, UMP, TMP. This is what we are going to make. That means we are going to make nucleotides, right? And if we make nucleotides, if we attach two nucleotides to like back, we are going to form one chain of the DNA, and complementary will have other chain of DNA, and that's how we are making DNA. So I'm going to talk about how we are making DNA. Okay, we know what is nucleoside, we know what is nucleotide. Let's learn how we make them. Okay, this is the whole perspective of what am I going to talk today. Now let's choose that first. We'll talk about pyrimidine production because that's kind of more challenging. Okay, you have more ATP left now just after we have passed just 10 minutes. So write down heading in your note that we are going to talk about pyrimidine synthesis. Okay, try to make as nice notes as possible and write whatever I'm saying. Okay, we are going to talk about pyrimidine synthesis. Now remember, in pyrimidine synthesis, we are going to form nitrogenous base first, and then we'll add sugar. And also keep your first aid beside you. Okay, and keep looking that diagram which is of purine and pyrimidine synthesis. First we'll make nitrogenous base and then we'll add sugar. In contrast to purine synthesis, we will make sugar first and then add nitrogenous base. Okay. Now, if you want to start pyrimidine synthesis, and finally our end product should be C, U, and T. Okay. That's our end product. That's what we want to make in pyrimidine synthesis. Let's start. You can also use the reference of first aid. Whatever you don't know, you can read that and tell it to me. Okay. Let's start. Um, and I will also have that in front of me if I forget the things. So we are going to start with glutamine. This is not something I want you to remember. Plus carbon dioxide. Okay, I'm just writing it down because this carbon dioxide name will help us in the next product. The next enzyme, carbon dioxide and glutamine, and we are combining carbon dioxide and glutamine in presence of an extremely important enzyme which is called as just called, does anybody know what is this enzyme? Carbamyl
Phosphate
phosphate.
2.
What is S?
Synthase.
Synthetase 2. Now there is a little difference between synthase and synthetase. If it uses ATP, we name that enzyme as synthetase. If it doesn't use ATP and you are making something, that's called as synthase. Okay. So here, if I said this is synthetase, that means clearly it will use some of the ATP. Okay. Forget about how many ATP it is using. Now, remember this. This is very high yield enzyme. Okay. Put a star mark here. Carbamoyl phosphate synthetase 2. Okay, this is the first important enzyme you want to remember. Now, if the name says carbamyl phosphate synthetase 2, that means there should be somewhere in the biochemistry carbamyl phosphate synthetase 1, and you need to be crystal clear about the differences. CPS2 is present in cytosol. Okay, so remember, CPS2 is present in cytosol. Do they want to test you that which enzyme is present in cytosol and mitochondria? Absolutely yes. That's USMLE all about. Okay. You want to know about urea cycle that's happening in mitochondria and cytoplasm both. You want to know about hemoglobin synthesis that's happening. First part is happening in mitochondria, middle part in cytoplasm, end part in again in mitochondria. You want to remember about ketone synthesis, okay? Mitochondria, cytosol. So you want to absolutely know where enzyme is present in cytoplasm or mitochondria. This is in cytoplasm, okay? CPS2 in cytoplasm, CPS1 is present in mitochondria. Okay, so put a side note somewhere, CPS1 in mitochondria, carbamyl phosphate synthetase 1 in mitochondria. And where is it used? Where is CPS1 used? Anybody?
Urea cycle.
Very good. It is used in urea cycle. So now you know CPS1, it's a problem of urea cycle. Okay. CPS2, it's the problem of what?
Pyrimidine synthesis.
Pyrimidine synthesis. Yes. CPS1 is the problem of urea cycle, problem in mitochondria. CPS2 is the problem of pyrimidine synthesis, problem of cytoplasm. Okay, very good, very good, very good. Okay, now if I said that this is carbamyl phosphate synthetase 2, what is it going to make by combining glutamine and carbon dioxide?
Carbamyl phosphate.
Thank God, biochemistry is little easy. Carbamyl phosphate. Okay. Now, don't confuse glutamine with glutamate. Okay. Glutamate, when you combine glutamate and ammonia, you are going to make glutamine. Okay. So, what's the difference between glutamine and glutamate? Glutamate, you add ammonia, you make glutamine. Okay. Perfect. So that's your carbamyl phosphate. After you form your carbamyl phosphate, we want to go towards next step, and we want to add aspartate. Okay, we want to add aspartate, and finally we want to make orotic acid. Okay, it's the same flow diagram which is present in your book. Carbamyl phosphate, you add aspartate, and you want to make orotic acid. Now, orotic acid, what is it? Is it a nitrogenous base or sugar? It's a nitrogenous base. Remember, in pyrimidine synthesis, this is what we begin with. We want to make nitrogenous base first, and then we'll add sugar, then we'll add phosphate, and then we'll make nucleotide. Okay, orotic acid is a nitrogenous base, so we made a nitrogenous base for pyrimidine synthesis. Now we'll add sugar, but before that there is a very important enzyme which helps in combination, in combining carbamyl phosphate and aspartate. Does anybody know enzyme you can cheat with your first aid? Carbamyl phosphate and aspartate, that's a very very important enzyme here. I'm going to write it with red color. And dihydroorotic acid dehydrogenase. Brr, that's a funny name, right? At least you can make somewhat meaning of this DHODH in your regional language. So let's dihydroorotate dehydrogenase. With the help of this enzyme, you can make orotic acid. Why am I making so much fuss about this? Because there is a drug which can inhibit this enzyme. Anybody? One drug which can inhibit this enzyme.
Leflunomide.
Very good. Leflunomide. Okay, we are going to talk about two drugs which can block in pyrimidine synthesis. Okay, so this is our first drug which we talked about. Leflunomide will inhibit dihydroorotate dehydrogenase. That means it's going to block the carbamyl phosphate and aspartate conversion into orotic acid. Okay, Simple, simple, simple. And pyrimidine synthesis, we knocked off our first drug. It inhibits dihydroorotate dehydrogenase. Now, you might not be asked about like where is leflunomide used, but tell me, what's the first line treatment of rheumatoid arthritis? First line treatment of rheumatoid arthritis. Rheumatoid arthritis, first line treatment.
Hydrochloroquine, glucocorticoids,
Um, Glucocorticoids, you can start glucocorticoids if the patient is having acute flare of rheumatoid arthritis, but that's not disease modifying.
DMARDs,
uh, Disease modifying anti-rheumatoid arthritis drug. What are the name of those drugs? Number one of rheumatoid arthritis starts from M.
Methotrexate.
Yes, methotrexate is the number one drug of choice for rheumatoid arthritis. Okay, please remember this. You start this drug right away. You don't wait. You don't want to wait with NSAIDs and steroids. You want to start this drug right away because that's disease modifying. Okay, number one is methotrexate. Now somewhere down the second line, third line, if that's methotrexate is not working, you may use leflunomide. Okay, so I'm writing this dot dot dot. You may somewhere use leflunomide. Okay, everything crystal clear until now? Perfect. Now, as I promised, we are going to make nitrogenous base first and then we'll add sugar. We are in that point right now. So let me tell you, where is that sugary thing coming from? So we know one basic thing. Sugar has to be ribose 5 phosphate. And you know now the source, it's going to be HMP shunt or PPP, okay, which is going to make ribose 5 phosphate. Now, everybody needs charging up, okay. Ribose 5 phosphate is kind of a sugar, but it's not ready to date yet. So if you are going for your date um, in evening, you got to order some dress with some high heels, some makeup, okay, whatever. So at least you just want to be charged up. So that's what ribose 5 phosphate is looking for. You want to get charged up before going to your date. So how is it going to be charged up? Now, in biochemistry, we are just studying biochemistry, and unfortunately, this is it. So in biochemistry, what's the date? If you want to charge up anything, you bump them with phosphate. Okay. So you just want to add phosphate to ribose 5 phosphate, and then phosphate in ribose 5 phosphate will be charged up. So the way to do that, the name of the enzyme is very easy. Okay. So the name of the enzyme which can does charging process. This is the second high yield, third high yield enzyme I'm talking about. Okay. So keep writing this. So our first high yield enzyme was CPS2. Then our second high yield enzyme was dihydroorotate dehydrogenase. And this is the third high yield enzyme which I'm going to write, just called as PRPP. Very high yield. Very high yield. Okay, PRPP. The name is very simple. Look at this. Phospho, phospho, ribosyl, pyrophosphate. And wherever you hear the word pyrophosphate, that means it's adding 2-phosphate. Pyrophosphate, that's just fire. Okay, it's lightening up ribose 5-phosphate. So phospho, ribosyl, pyrophosphate synthetase, PRPP synthetase. Okay, so I'm gonna combine these. So ribose 5 phosphate, you want to add 2 more phosphate in ribose 5 phosphate with the help of enzyme PRPP synthetase. Now, as the name says, it's PRPP synthetase, and you will make what will be your end product? Just PRPP. Okay, keep writing, keep on the track. So PRPP. Now tell me, what is PRPP? Out of the three things which I talked about, is it a phosphate? Is it a nucleotide? Is it a sugar? Is it a nitrogenous base? What is it?
Sugar.
Yes. Thank you. It's a sugar. Very good. Okay. It's the sugar. And what is orotic acid again?
Nitrogenous base.
Nitrogenous base. Now I'm going to combine those. Okay. So orotic acid is now going to combine with PRPP. So just guess what? What is it going to form? Orotic acid is a nitrogenous base. We have a sugar. Does it have phosphate as well? Does PRPP have phosphate?
No.
Oh, yes, it does. It has two phosphate. Now it has actually three phosphate. The ribose 5 phosphate already had phosphate. Now if you are making pyrophosphate means addition of two more phosphate. Okay, so it's having a lot of phosphate. Trust me. PRPP is having a lot of phosphate. Phosphoribosyl pyrophosphate. It is having phosphate. So that's sugar with phosphate, and we have nitrogenous base. So guess what? What is it going to make?
Nucleotide.
Nucleotide. So whatever product is going to be here, it's going to be a nucleotide, right? And the nucleotide first, we formed our first nucleotide, guys. Congratulations. And that's going to be UMP, uridine monophosphate. Okay, we made our first nucleotide. Remember, we are in pyrimidine synthesis, so we wanted to make, out of three, we wanted to make CUT. So we made U, okay, uridine monophosphate. Perfect. Another um, important enzyme here. Any guesses? If you want to make UMP, what enzyme will you make? I'm asking this because it's simple. You want to synthesize UMP, what enzyme should you use?
UMP synthesis.
Perfect. That's UMP synthase here. It's whatever you want to use. Synthase doesn't make any sense for medical students, but it does make sense for biochemists. Okay, they will kick us if we use synthase and synthetase interchangeably. But anyways, just let's just keep it UMP synthase, whatever you want to say. It's UMP synthase here. Okay, that's our fourth important enzyme, guys. UMP synthase. And the reason why this is important, because imagine, can you tell me what will happen if I have deficiency of UMP synthase? What levels will go up?
Orotic acid.
Yes. UMP synthase deficiency will lead to orotic acid urea. Isn't that high yield? We are going to go slow, but you are going to learn each and every important thing here. So number one thing we learned, UMP synthase deficiency. If you have deficiency of this, your orotic acid will be high in your blood, and this orotic acid will go into the urine. So UMP synthase deficiency will give you orotic acid urea. Guys, this is very important because we'll talk about orotic acid urea at three places, and this is the first place which I'm talking about, UMP synthase deficiency, and that's an enzyme deficiency. Okay. So UMP synthase deficiency, I'm going to write down here, that's it's impaired in orotic acid urea. Okay, very, very important. That's our enzyme number 4. Perfect. Let's move on. Now, this is going to be easy. You made uridine monophosphate, right? You made uridine monophosphate. There is no difficulty in saying that I can convert uridine monophosphate to uridine diphosphate. Okay, you just add one more phosphate. That's UDP. Now, if I want to convert UDP into, now it has two pathways. Okay, first thing is you can shift your gears. You can somehow, with some magic, okay, UDP is converted into C. So we made some cytidine as well. Okay, so you're going to make, let's say with some magic, you're going to make cytidine mono, di, and triphosphate. Okay, so that's cytidine. So look here, we made already two nucleotides here. I'm going to hide the highlight here for you. UDP is our first product. We also made your CTP. Okay, so CTP. We already made two nucleotides. Now, simple thing. Uridine diphosphate. Uridine diphosphate. That's a nucleotide. A nucleotide with a ribose sugar in it. Okay, so I can say this is a ribonucleotide. Correct? Uridine diphosphate. It's a nucleotide. Correct? And the sugar which I used was ribose 5-phosphate. So I can say this is a ribonucleotide. And if I want to reduce this ribonucleotide, that means reduction means either you add hydrogen or you remove oxygen. That means I want to reduce this UDP and make deoxy UDP. So that's a reduction reaction. So I'm going to use enzyme which can make UDP into deoxy UDP, and this enzyme is called as ribonucleotide reductase. Your 1, 2, 3, 4, fifth high yield enzyme. Okay, so write down here ribonucleotide reductase. The reason why this is important, because we have drugs acting on this enzyme. Can you tell me one drug which can inhibit ribonucleotide reductase? And this is going to be our second check mark.
Hydroxyurea.
Hydroxyurea.
Hydroxyurea, very good. Hydroxyurea. Hydroxyurea is your second drug here, which can block ribonucleotide reductase. So in pyrimidine synthesis, we have hydroxyurea, we have leflunomide. So actually this is kind of a common between pyrimidine and purine synthesis, so you can just name it as a common drug as well. Okay, hydroxyurea blocking ribonucleotide reductase. Now tell me, where is hydroxyurea used?
Sickle cell anemia.
Very good. Hydroxyurea is used in sickle cell anemia. And what does it do?
They will increase fetal hemoglobin.
Excellent, Babita. Very good. Hydroxyurea is going to increase the fetal hemoglobin. Now tell me, I'll be really impressed. What's the composition of fetal hemoglobin? Alpha, beta, gamma, delta, what?
Alpha and gamma or delta?
No,
Beta.
it's uh, uh, hemoglobin F is
Alpha and
alpha 2.
delta.
Beta 2.
No. Alpha 2 and gamma 2. Hemoglobin F
Okay.
is alpha 2 and gamma 2. Hemoglobin A2 is
Alpha and delta.
alpha 2 delta 2. And what are we left to with? The hemoglobin. So we have fetal hemoglobin, hemoglobin A2, and
Adult hemoglobin.
HbA1, which is
Alpha 2, beta 2.
alpha 2 beta 2. Perfect. Okay. So we have alpha 2 beta 2, alpha 2 delta 2, and HbF, that's alpha 2 gamma 2. So in hydroxyurea sickle cell anemia, you are going to increase your HbF, because in sickle cell anemia you don't have enough of hemoglobin, so you want to somehow increase, that's hemoglobin S. In sickle cell anemia you want to increase HbF with help of hydroxyurea. Now look at this. If you give hydroxyurea, you will not be able to make deoxy UDP. So this is all going to back up. Okay, this is all going to back up. Okay, That's your hydroxyurea. So right now we are in deoxy UDP stage. One another fact you want to note down in your chart here is that if somebody has orotic aciduria, you cannot make uridine, correct? If you have orotic aciduria, you cannot make uridine. So if somebody has deficiency of UMP synthase, your question will be which of the following supplements can help to overcome this deficiency? What will be your answer?
B12.
Um,
B2.
why B12?
Uridine.
Uridine. Correct. See, you are not able to make uridine, so give uridine. Correct. So supplement uridine. So in this chart, you want to understand that if you have a certain deficiency, if you're not able to make something, just supplement that. If you're not able to make uridine, supplement uridine. So your supplement in uridine, orotic aciduria, is going to be uridine. Okay, very high yield concept. Okay, now, so this is your pyrimidine synthesis. We made UDP, we made CTP. Now I'm going to write a mini version here. Just continue in your chart in just parallel fashion. Okay, I might just switch to another board because I don't have space here. I like this. I don't want to erase this. Oop, very good. So what I'm going to write down a mini version here, that let's say you have DUDP, that's deoxy UDP. Of course, you can convert that into deoxy uridine monophosphate, dump deoxy uridine monophosphate, and then finally with some magic, we will convert on the next page this deoxy UMP into deoxy TMP. Okay, deoxy TMP. So make sure you have some space here, because in next page I'm going to explain how you can convert deoxy UMP into deoxy TMP. So look at this pyrimidine synthesis. We wanted to make pyrimidine, right? So we made our first pyrimidine, that's nucleotide, uridine diphosphate. Then we made cytidine triphosphate. Then we also made deoxythymidine um, monophosphate. Uridine is only going to be present in RNA. In contrast, you can use cytosine and thymine in your DNA, right? Perfect. Now, any questions on this slide before we jump on to the next page? Okay. You also want to remember and write this down, that leflunomide can target S phase of your cell cycle, and hydroxyurea can also target S phase of your cell cycle. Okay, just write that down. Perfect. Now here I'm going to zoom in. Okay, I'm going to zoom in this on the next page. DUMP to DTMP conversion. So I'm going to just start off with DUMP, dump. Okay, and this slide is going to be extremely important. Deoxyuridine monophosphate. We want to somehow convert this into deoxythymidine monophosphate. We want to convert dump into DTMP. Okay, and this happens with the help of one very important vitamin. And what is that? Folate. Folate. Remember this cut, CUT. I told you that if you deaminate cytosine, you make uracil. But if you do something to uracil, you make thymine. What was that something? You want to add methyl group. Okay, if you add methyl group to dump, you will make DTMP. So somehow I want to donate uracil, a methyl group. In biochemistry, as yesterday I told you, there are only two carriers of methyl group, two carriers of methyl. Okay, so number one is going to be your folate, and number two is going to be, who's the second methyl donor, or also called as carbon donor? S. Adenosine?
I say M.
Yes, as adenosine, methionine. Okay, these are two carbon donor. So whenever I say next time, do you know one carbon reaction in biochemistry? Say me yes. This is one carbon reaction. I'm donating one carbon here. Okay, there are two carbon donors in biochemistry we want to learn about. One is folate, which is a methyl donor, and second one is SAM. Okay, perfect. Now, the way by which folate can donate its carbon or methyl, it has to have carbon by itself, right? So the type of folate which has carbon is called as methylene, has methyl group. Methylene, there's a difference between methyl and methylene. Methylene tetrahydrofolate. So this has a crown. Ooh, okay. This crown is methyl. So methylene tetrahydrofolate has methyl. So if you want to donate this, you can donate it here. Okay. So methylene tetrahydrofolate will donate methyl group to DUMP, and this DUMP can convert into DTMP in presence of a very important enzyme, which is called as, we want to make thymine. Thymine. We want to synthesize thymine. So enzyme has to be something like thymine synthase. So actually it's thymidylate synthase. Okay. Thymidylate synthase, another high yield enzyme. Ooh, thymidylate synthase. Okay. So just write this down and encircle. This is very important enzyme because we have a drug which can inhibit that enzyme. And what is that drug?
5-fluorouracil.
Excellent. 5-fluorouracil can inhibit thymidylate synthase. And there is one prodrug of 5-fluorouracil, which I'm not going to write that down because I don't like that drug, the name of that drug. Capecitabine. Okay. Capecitabine is a prodrug of 5-fluorouracil. So I'm not going to write that down. But you can go ahead and write that down. Okay, capecitabine is a prodrug of 5-fluorouracil that's similarly in mechanism of action going to inhibit thymidylate synthase. Okay, tell me, what's flucytosine? Have you heard of flucytosine? Where is it used? Phi flucytosine. Flucytosine. Have you heard of this treatment somewhere? You combine flucytosine with amphotericin B, you treat one of the infection, followed by maintenance therapy with fluconazole. Does it sound and ring some bell? Cryptococcal meningitis. So drug of choice for cryptococcal meningitis is you combine amphotericin B with flucytosine followed by maintenance therapy. You remember that sketchy diagram? Flucytosine, amphotericin B, there were like frogs followed by azoles. That's fluconazole, okay? That's the treatment of cryptococcal meningitis. Remember those India ink stain and all those stuff? Latex agglutination, um, cryptococcal meningitis spread by pigeons, and you had that red music carmine red stain. um, Cryptococcal meningitis. So that flu cytosine, how does it act? Okay, so that flu cytosine, which is a treatment in cryptococcal, it's actually 5-flu cytosine. Okay, that 5-flu cytosine can convert into 5-fluorouracil. Okay, we have an enzyme which you don't need to remember, and that's cytosine deaminase, which helps the conversion of 5-fluorouracil to 5-flu cytosine to 5-fluorouracil, and that 5-fluorouracil can inhibit thymidylate synthase, and you cannot make a nucleotide. You cannot make thymidine. If you cannot make a nucleotide, that means you cannot make your DNA. That means that organism has to die, right? That's how those antifungal is working. Okay, perfect. So now you learned the mechanism of 5-fluorouracil, and it's a drug, capecitabine. Okay, so if you give this drug, that will decrease the production of DTMP. Perfect. Now, what will happen to methylene tetrahydrofolate? If you are donating methyl group to DUMP, it itself is going to be oxidized or reduced. If you are donating hydrogen to somebody, what will happen to yourself?
Reductase,
Um,
mm, losing oxygen, so it's not
losing oxygen
reduction.
is reduction, gaining hydrogen is reduction, but giving hydrogen is oxidation. Okay,
Oh,
so forget about it. Tetrahydrofolate, you are just donating um, methyl group, and you're also donating some hydrogen. So that tetrahydrofolate will convert into dihydrofolate. That's oxidation reaction. Okay, so if I say this is oxidation reaction, if you somehow want to convert dihydrofolate into tetrahydrofolate, it has to be a reduction reaction. Do you guys agree? If you want to convert dihydro into tetrahydro, from 2 hydrogen to 4 hydrogen, you are adding more hydrogen. It has to be an reduction reaction. So there has to be some reduction enzyme here. Okay, so if you want to convert dihydrofolate into tetrahydrofolate, that means it's a reduction reaction, and tetrahydrofolate can eventually convert into methylene tetrahydrofolate. You want to again donate one methyl group from somewhere that comes from one of the amino acid, with the conversion of serine to glycine, which you don't need to remember. Okay, so you may not write this down. So just write this down: dihydrofolate to tetrahydrofolate. We want to reduce dihydrofolate into tetrahydrofolate. What enzyme? DHF to THF.
dihydrofolate reductase.
Very good. Dihydrofolate reductase. So that's an enzyme which can help to reduce DHF into THF. Okay. Very important. Ooh, another very important enzyme, because we have drugs which can inhibit this enzyme depending on the species, depending on whether you are prokaryote, eukaryote, or protozoa. Tell me a drug which can inhibit prokaryotic dihydrofolate reductase. If you are prokaryote, eukaryote, or protozoa. Okay, tell me any drug you know which can inhibit dihydrofolate reductase.
And
Methotrexate,
methotrexate.
Methotrexate. So methotrexate, we are using methotrexate, right? That's we are using that in rheumatoid arthritis, but that has to be eukaryotic. So methotrexate is a drug which can block eukaryotic dihydrofolate reductase. Okay, so write methotrexate here. Methotrexate blocking eukaryotic dihydrofolate reductase. Methotrexate. One of the side effect of methotrexate is what? You can use this diagram.
pulmonary fibrosis,
Okay, pulmonary fibrosis is one of the side effect of methotrexate. What else? What
bone
deficiency?
marrow suppression,
Bone marrow suppression. Why?
uh, because DNA, sorry, thymine
Yes.
won't synthesize.
Yes, because you are not making thymine. And if you are not making thymine, you cannot make DNA properly, and your hematopoietic cell want to rapidly multiply, and they cannot use thymine, so they cannot make DNA, so they will not be able to multiply. So bone marrow suppression means pancytopenia, right? So how can you overcome the side effect of methotrexate? And if you cannot make DNA, that's sort of a megaloblastic anemia, right? How can you?
Leucovorin.
Leucovorin. Perfect. So if you want to alleviate the toxicity of methotrexate, you can rescue with leucovorin. Leucovorin. Okay, extremely important. You can rescue with leucovorin. What is leucovorin?
Folinic acid.
That's a folinic acid. So is it dihydrofolate or tetrahydrofolate? It's a
Dihydro.
It is if you give dihydrofolate, if let's say this is usual folic acid, if you just take folic acid as your supplement, normal folic acid, that's probably dihydrofolate, because we have enzyme dihydrofolate reductase functional, or if you are not taking the drug, you can convert dihydrofolate into tetrahydrofolate and you can use that in nucleotide synthesis. But if the patient is taking this drug, you cannot give them dihydrofolate because you are inhibiting this enzyme. So you should give either this or you should give either this. So it has to be reduced dihydrofolate, which is either tetrahydrofolate or methylene tetrahydrofolate. Does it make sense, Yashasvi?
Yes, sir.
Very good. So it has to be reduced. This one, it's a reduced folate. Leucovorin. It's not an, if you just go over the counter, take folic acid, that's not the folic acid you want to give to this patient. You want to give them folinic acid, which is a reduced form of folate, tetrahydrofolate. Okay. Very important. So you can rescue methotrexate toxicity with leucovorin, which is also called as folinic acid. Folinic acid, which is also called as reduced tetrahydrofolate. Reduced folate. Okay. Uh, you also want to remember methotrexate will also block the S phase of the cell cycle, synthesis phase. Now, there are some other drugs which can block dihydrofolate reductase, like for example, TMP, trimethoprim. If you combine that with sulfamethoxazole, we call that as TMP-SMX. Okay. So TMP, I'm writing TMP-SMX in the bracket, TMP-SMX in the bracket. TMP-SMX, so TMP-SMX would block the prokaryotic dihydrofolate reductase, extremely high yield. Okay. So folate, bacteria will not able to make folate and they'll not be able to make DNA. Can you tell me where do we use TMP-SMX? Bactrim, which is also called as Bactrim in hospital. Where do we use?
In case of toxoplasmosis, no, I think that was different.
Um, Toxoplasma. So is it
Something
used?
is like this.
So is it used in the treatment of toxoplasmosis or the prophylaxis of toxoplasmosis?
Prophylaxis.
Prophylaxis. You are correct. Right. So prophylaxis. Prophylaxis. Where do we want to give prophylactic dose of TMP-SMX for toxoplasma? You remember this three things: PTM, PTM. If CD4 count are less than 200, we want to give them pneumocystis cerevisiae pneumonia, PJP or PCP pneumonia prophylaxis. That's TMP SMX. If the CD4 count are less than 100, we want to give them toxoplasma prophylaxis, which is also TMP SMX. And if the CD4 count are less than 50, we want to give them MAC prophylaxis. What is MAC? Mycobacterium avium complex. And we want to give them azithromycin here, but the guideline is changed here. So these are CD, okay, CD4. So I'm talking about PJP prophylaxis, where you can use, I already gave the answer. So you said toxoplasma, where we can also use TMP-SMX, but we can also use prophylactic in PJP. So for PJP pneumonia or pneumocystis cerevisiae pneumonia or pneumocystis carinii, it was called as PCP. In PJP pneumonia, you can use both prophylactic TMP-SMX and treatment of TMP-SMX. Okay. So TMP-SMX can be used as both prophylactic and treatment for PJP and only prophylactic for toxoplasma. Okay. Where else we can use TMP-SMX? Urinary tract infection, right? Along with nitrofurantoin, Bactrim or TMP-SMX is a first line drug of choice for urinary tract infection. Okay. So if I said that you can use prophylactic TMP-SMX for toxo, and for pneumocystis cerevisiae, you can use TMP-SMX both for treatment and prophylaxis. Can you tell me what's the treatment of toxoplasma? Have you heard of pyrimethamine? Pyrimethamine. Pyrimethamine. So this is trimethoprim sulfamethoxazole. Pyrimethamine. Pyrimethamine along with the sulfur drug. You are going to treat toxoplasma. Very important. Okay, so remember we have two different drugs for the prophylaxis and the treatment of toxoplasma. If you want to give prophylaxis for toxoplasma, give TMPSMX. If you want to treat, you want to give pyrimethamine along with sulfadiazine. Okay, wonderful, wonderful. Any questions? Okay, so we now know drugs which can block dihydrofolate reductase. We now know drugs which can block 5 thymidinate synthase. What else I want to talk about? There is a thing that if you give folate, if the patient is taking methotrexate, that can reduce the toxicity of methotrexate, right? But if you give folate in patients who are taking 5-fluorouracil, you may increase the toxicity of 5-fluorouracil. The reason is if you give folinic acid, that's going to combine strongly with DUMP, and that can combine strongly with thymidylate synthase, and that can be more inhibited by 5-fluorouracil. So that's kind of a reverse. Okay, so I'm going to write down a small mini note here that is a gunner level thing. If you give folinic acid, okay, that may reduce toxicity if the patient is taking methotrexate, but it may increase the toxicity. In other words, you can say it can increase the efficacy for 5-fluorouracil. Okay, very important. Okay, if I say somebody has um deficiency of folate, what thing you can give as a supplement that you might not need folate at all? Let's say this entire step is not working. What supplementation you can give to the patient if your entire step is not working? Just give them if they want to make their DNA. Thymidine. Okay, so give them thymidine. So I talked about 3 supplements today. If the patient has UMP synthase deficiency, you want to supplement them with? Oh, looks like ATP is down. UMP synthase deficiency, supplement them with?
Uridine.
Very good. Methotrexate toxicity, supplement them with?
Folate.
Folinic acid. And then finally, if you don't want to have folate, what do you want to supplement?
Thymidine.
Thymidine. Wonderful. Any questions about pyrimidine synthesis? Excellent. So this is your pyrimidine synthesis.